Think that you have become a very powerful person and now, no force can defeat you. You are free from all diseases. Stop thinking, because now science has progressed so much that you can modify any living thing at will. In today's video I am gonna tell you about a very interesting biotechnology technique, which is genetic engineering. In this video we will discuss about genetic engineering, the whole process, applications and future of genetic engineering. Let's go. Genetic engineering is the process of manipulating an organism's genetic material, DNA, using biotechnology techniques to introduce new traits or characteristics. This can involve modifying an organism's existing genetic material, introducing new genetic material from another organisms, or deleting or suppressing unwanted genes. Genetic engineering can be used to create crops that are more resistant to pests or environmental stress, to produce medicines and vaccines, to develop new biotechnological products, and to study the function of specific genes. Now let's discuss about the whole process and technique of genetic engineering step by step, easy way to understand. Step number 1, the first step in genetic engineering is to isolate the DNA that you want to be modified. This can be done by extracting DNA from cells or by synthesizing DNA in a laboratory. Step number 2, once the DNA has been isolated, it needs to be cut into smaller fragments using enzymes called restriction enzymes. These enzymes recognize specific sequences of DNA and cut the DNA at those points. Step number 3, to insert the new DNA into the cut DNA. This can be done by using a variety of techniques, such as using a plasmid to transfer the new DNA into the host cell. Step number 4, the modified DNA needs to be transferred into the host cell. This is done by using electroporation or chemical transformation techniques. Step number 5, once the modified DNA has been transferred into the host cell, the next step is to select for the desired trait. This is done by growing cell into specific environment or by using a selection marker to identify the cells that have been successfully transformed. Step number 6. Once the cell have been selected, the modified DNA needs to be characterized to ensure that it has been modified as desired. This can be done by using PCR, gel electrophoresis or DNA sequencing techniques. Step number 7. Once the modified DNA has been characterized, it can be scaled up for production. This involves growing the cells in large bioreactors and harvesting the modified DNA. Our whole process has completed now. Let's talk about applications of genetic engineering. Number 1, it is used in agriculture area to create crops that are resistant to pest, diseases, and environmental stress. Number 2, it is used in medical purpose such as insulin and growth hormone, that are used to treat diseases such as diabetes, and growth disorders. Number 3, in industrial biotechnology, it is used to produce enzymes, chemicals, and other products that are used in variety of industries, such as production of biofuels, bioplastic and pharmaceuticals. Number 4, to create organism that can biodegrade pollutants or remove toxins from the environment. Number 5, genetic engineering is used to forensics to analyze DNA evidence and identify suspects in criminal investigations. Number 6, it is used in welfare of livestock, as well as to create genetically modified animals for research purposes. Now let's discuss about future aspects of genetic engineering, which is very important to understand yet. The future of genetic engineering includes gene editing, synthetic biology, personalized medicine, gene drives, agricultural advancements, and human enhancement. These advancements have the potential to cure genetic diseases, create new materials, eradicate disease-carrying new materials, eradicate disease-carrying insects, improve crop yields and nutritional value, and enhance human traits. However, there are ethical concerns regarding unintended consequences, misuse, eugenics, and inequality. I hope you all enjoying this video and get informative. Subscribe, please.